Okay, so in today's lecture, what we're going to be doing is we're going to be moving away from the cellular bases of neuroscience, where we've already talked about neurotransmitter systems, we talked about synaptic transmission, we talked about resting membrane potentials and action potentials. Now we're going to be looking at the nervous system, especially in this chapter, as a whole. So the structure, the overall structure of the nervous system, the divisions of the nervous system, and the development of the nervous system. So the nervous system is really, well, it's the entire study of neuroscience, right? So the structure of the nervous system tells us about brain functioning, and when we have damage to the nervous system, you're able to really see the deficits that are a result of damage to that structure. So brain organization follows the mammalian plan, where despite differences in the amount of complexity that our brains show, the brains from numerous different species all share common features. And here you can see general shared features such as cerebrums in dolphins, humans, chimpanzees, sheep, rabbits, olfactory bulbs here. So in humans, the olfactory bulb's like on the ventral side, and it's very, 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 very small. But notice that in a rat and in a rabbit, these take up a significant amount of the actual brain, right? And that's important because rats and rabbits, they spend a lot of time sniffing. Same thing with cats as well. So the nervous system can be divided into two major divisions. You have the central nervous system and the peripheral nervous system. The central nervous system is composed of the brain and spinal cord, and that's it. So if you were to look at a person right here with its hands and feet, the central nervous system lies along that central axis where the brain and spinal cord are. And then the peripheral nervous system is everything outside of that. So the nerves that are sending information out towards the muscles and to the glands, and then nerves that are sending information from the skin and from the glands and tissue to the central nervous system. So in neuroscience, it's really important to understand anatomical references. So if I were to ask either of you or any of you directions to a particular location, you wouldn't just tell me it's in the center of campus or, it's to the left. In order to get around in the nervous system, we have to use directional landmarks. So if we refer to something as anterior or rostral, we're saying that something is in front of something else. If something is posterior or caudal, it's behind something. Dorsal is towards the top, whereas ventral is towards the bottom. Medial is toward the midline. Lateral is going out to the side. Ipsilateral is within the same side, and contralateral is on the opposite side. And as we discuss the sensory systems in the next few lectures, we'll be using some of these terms more often, in particular with ipsilateral and contralateral projections in some of the sensory systems. So being able to understand that ipsilateral is connections or are connections that remain on the same side of the body, whereas contralateral projections are projections that start on one side and go to the other, that's going to be very important to understand and know. As well as the fact that when we're talking about where something is, we're going to be using these anatomical references, so it's important to understand them now. So here, dorsal is towards the top. So if you think about a shark or a fish, like with a dolphin, a dorsal fin lies on the top. A ventral is towards the stomach. And so in an animal that lies on all 4 feet, there's a parallel sense to the brain and spinal cord, right? However, we humans, we like to walk. So there's this axis that goes down when we're talking and when we are operating, where the spinal cord, dorsal side, is out here, but the brain, dorsal side, is up here. Whereas in an animal, this is all dorsal. So we have the brain, the spinal cord, which lies down here. This is the midline of the animal. Lateral is out to the side, medial is towards the midline. And then ipsilateral is when 2 structures are on the same side, and contralateral is when 2 structures are on opposite sides of the midline. So let's say right here and right here, these are contralaterally to each other. So let's call this A and B. A and B are contralateral to each other because they lie across the midline, whereas A and C are ipsilateral structures because they lie on the same side. The brain can also be sectioned into different planes of section. You have the mid-sagittal plane, which splits the brain into two equal halves, the left and the right. This is along the medial longitudinal fissure. Now, this is a mid-sagittal because it is technically a sagittal cut, but in the midline of the brain. The sagittal plane are sections that lie parallel to this mid-sagittal plane. So imagine a cut that's going right here, going down exactly the same way. Horizontal plane, think about the horizon. So if you think about a landscape and then a horizon being the sun. This is parallel to the ground, and it splits the brains into a dorsal half and then a ventral half, which is down here. A ventral half and a dorsal half. And then a coronal plane lies perpendicular to the ground. So if the ground lies right here, a coronal plane is going to be perpendicular to that. This is going to split the brain into anterior, which means forward parts, and posterior parts. This is the plane of section that I slice brains in. I slice brains in coronal sections. And if you take 373 or if you take any kind of anatomy lab, you'll be able to see all these different structures in different views based on how you actually slice the tissue. So the overall organization of the nervous system, the central nervous system are the parts of the nervous system that are encased in bone. You have the skull and the bony vertebral column. The cerebrum, cerebellum, and brainstem are common to all mammals. However, it's just the complexity, particularly of the cerebrum, that really gives humans their advanced capabilities. So the central nervous system is made up of the brain and spinal cord. So cerebrum, cerebellum, brainstem, this is the brain, right? So the cerebrum is divided into two hemispheres, and here it's found in blue. So each controls sensations and movements of the opposite side of the body. The cerebellum, right here, is a movement control center with numerous connections with the cerebrum and spinal cord. And the brainstem, which is here, is the site where vital functions are going to be regulated, including breathing, consciousness, and the control of body temperature. You really don't want damage to the brainstem. If you have damage to the brainstem, most likely death is going to occur due to the fact that there are so many autonomic functions that are controlled at the level of the brainstem. Now, the spinal cord is attached to the brainstem and encased in the bony vertebral column, and it's a conduit of brain-body information. So you have information from skin, joints, and muscles that will go to the brain, and then information from the brain that will then go to joints and muscles and skin. And you'll learn a lot about these pathways in the next part of the class, which is Psych 376, Brain and Behavior, where you start off with the interactions of the nervous system and the muscle system. If you transect the spinal cord or if you cut the spinal cord, you cannot feel the skin and muscle paralysis caudal or below the cut. So here's a brain and spinal cord. If I were to cut the spinal cord here, I would lose the ability to move and I would lose all sensation from down here. Now the reason for that is because the brain is connected again to the spinal cord and it is unable to pass this level of transection, right? So it can't communicate with anything down here. And then anything down here can't send information up because, again, there's a level of transection. So the muscles literally cannot be controlled by the brain. And a little throwback to chapter 1, but the Bell-Magendie law, where we have dorsal sensory information that is coming into the nervous system, and then ventral roots carry motor information out. So these are classifications of spinal nerves. And here's the spinal cord looking at a caudal section. Here are the dorsal root ganglia, and ganglia is just a term for collection of cell bodies. So you have the dorsal roots, which are bringing in sensory information, and then you have exiting ventral roots, which are taking away motor information. And these collectively are referred to as spinal nerves. So we said that the nervous system is divided up into the central nervous system and the peripheral nervous system. Well, what are the further divisions of the peripheral nervous system? This is, again, the nervous system that lies outside of the brain and spinal cord. This is composed of the somatic peripheral nervous system, or the somatic nervous system, and the visceral nervous system, or the autonomic nervous system. So the somatic nervous system is controlling our voluntary control. So this is innervating skin, joints, muscles. We're able to move and react and bring in sensory information, particularly in the form of touch, into our nervous system for processing. So these are in the dorsal root ganglia. These are clusters of neuronal cell bodies that lie outside of the spinal cord, and these are containing those somatic sensory axons, bringing in information from the periphery. Whereas the autonomic nervous system innervates internal organs, blood vessels, and glands, and this is involuntary. So this is communicating critical information to the brain that you do not want conscious control of. So sensory fibers help to bring in information about pressure and oxygen content of blood, while motor fibers help to command contraction and relaxation of smooth muscles that you really don't want to have to be thinking about, right? So muscles in the gut, right? You don't really want to have to be consciously controlling contraction and relaxation of those interior autonomic muscles. So in the autonomic pathway of the peripheral nervous system, you have information that's going to go up through the dorsal root ganglia to the spinal cord, and then information that will be able to go out from the spinal cord back to the glands and to smooth muscles. In the somatic pathway, you have information from sensory neurons from, let's say, the skin going into the dorsal root ganglia for processing, and this information can then go upwards to the brain for cortical processing. And then information that leaves out of the ventral roots going to the skeletal muscle. Now, the somatic pathway is under voluntary control. The autonomic pathway, as the name suggests, is automatic. It's non-conscious. You're aware that it's happening, but you're not physically controlling its occurrence. So, afferent axons carry information towards a particular point. Efferent carry information away from. And it all deals with perspective. So something can be afferent to something else. But usually sensory neurons, or not usually, these are sensory neurons are afferent fibers. They are bringing information into the central nervous system. So think of A for accept. And efferent fibers carry information away from a point. So these are motor fibers in the spinal cord. These are efferent fibers which will then go out to the periphery to innervate a muscle or a gland. So remember when we talked about the Bell-Megendi law and we said that sensory is going to be dorsal and it's going to be afferent. That information is coming into the central nervous system. Those are afferent fibers. Visceral, or not visceral, but ventral motor information is going to be efferent. This is carrying information away from the central nervous system. There are 12 pairs of cranial nerves. Mostly they innervate the head, but some are axons from the central nervous system, some from the somatic peripheral nervous system or visceral peripheral nervous system, and some have strictly sensory motor, or some of them have both characteristics, both of sensory and motor. Now these are the 12 cranial nerves, and notice that all of them are important for our class. The only nerves not mentioned are the spinal accessory and the hypoglossal nerves, which are going to be discussed further in 376. But the important ones for our class are the olfactory nerve, which is cranial nerve 1. This is a sensory nerve and it's conveying the sense of smell. The optic nerve is cranial nerve 2, which is a sensory nerve carrying vision, or the ability of our eyes to bring in visual stimuli. The oculomotor nerve, which is cranial nerve 3, it's a motor nerve and it allows for eye movements along with the trochlear and the abducens nerve. The oculomotor, trochlear, and abducens all convey different degrees of eye movements. The trochlear is cranial nerve 4 and it has motor abilities. Abducens is cranial nerve 6 with motor abilities. The trigeminal nerve is the first cranial nerve that has components of both. So it conveys touch and pain sensation of the face, which are sensory, and then allows for muscles of chewing to occur or to work. Cranial nerve 7, 9, and 10, these are important when we start talking about our taste system. These convey taste information that will be able to go in from our sense of gustation and help to control the muscles in our throat. And then the vestibulocochlear nerve, which is cranial nerve 8, which has a sensory characteristic allowing for hearing and balance. Now, you won't have to memorize these for my class, but you will need to know them for 373 if you're taking physiological psychology lab. But for our class, you will need to be able to highlight the important ones as we go through each of the sensory systems. So when we talk about vision, you should really be familiar with the optic nerve. When we start talking about olfaction, you should be really experts at the olfactory nerve. When we also talk about vision, we'll talk about the oculomotor, the trochlear, and the abducens. When we talk about gustation and taste, we'll talk about 7, 9, and 10. When we talk about hearing and vestibular systems, we'll talk about the vestibular cochlear nerve. So you won't necessarily need to memorize them all, but you will need to know how each individual one works in its given sensory system. But the nerves not mentioned that I'll still talk about are the spinal accessory, which is a motor nerve, which conveys the movement of the head and neck, and the hypoglossal nerve, which conveys movement of the tongue. The meninges are 3 membranes that surround the brain. You have the dura mater, which is the tough outer layer, the arachnoid membrane, which has the subarachnoid space that's filled with our cerebral spinal fluid, and you have the pia mater, which is a thin membrane that actually lies adheres very closely to the surface of the brain. So if you look at this picture right here, this is what the brain actually looks like with its protective meningeal layer surrounding it. So your brain isn't just in your skull, not further protected. The meninges serve as additional membranous layers that help to surround and nourish and protect the brain. This is part of the ventricular system. So the ventricles are the cerebral spinal fluid-filled caverns and canals that lie within inside our brain. The choroid plexus is a specialized tissue in the ventricles that will secrete the cerebral spinal fluid, allowing for a continuous flow. And the cerebral spinal fluid circulates throughout our ventricles, and it's absorbed in the subarachnoid space for recycling. So by ways of small openings located near where the cerebellum attaches to the brainstem, we're going to be having a general circulatory system in regards to the ventricular system. So the cerebral spinal fluid is absorbed by the blood vessels at arachnoid villi, and brain damage and obstruction of any of this flow can actually result in hydrocephalus, which literally means water on the brain, where you can actually remedy this by the administration of shunts. So essentially, you can insert a tube into the lateral ventricle, which will be able to drain excess fluid down here into the peritoneal cavity. Now what that does is it allows for there to be less pressure on the brain. Because if you have a very young child that has increased pressure being pushed down onto the brain through the cerebral spinal fluid accumulation, then you might result in mental retardation or intellectual handicaps. So this video is a video from or a demonstration of the human ventricular system. And we'll see if this will work. Okay, guess not. But this demonstration just shows the way of cerebral spinal fluid flowing throughout the ventricular system. Here are the ventricles that lie within the brain. You have the lateral ventricles, the third ventricle, and the fourth ventricle. And as we get into the development of the nervous system, we'll talk a little bit more about where each of these are in regards to landmarks for other brain structures. So the other part of this chapter discusses some of the research methods that are used to visualize the brain. So we'll start with computerized tomography. And this helps to generate an image of a brain slice. Now what CT or CAT scans do is that they use X-rays that will allow for data to be essentially generated to construct a digital image. It's non-invasive, so nothing's actually going into the brain, albeit X-rays are. But this helps to reveal gross organization, and by gross I just mean overall, organization of gray and white matter and the position of the ventricles in the living brain. It's not functional. So not functional. Meaning that I can't put you in a CAT scan ask you to do a task and I would be able to see what area of the brain lights up. I can't do that. But it's really good for noting brain anatomy. Now, magnetic resonance imaging, or MRI, is based on how hydrogen atoms respond in the brain to perturbations of a strong magnetic field. Now, these signals are mapped by computers to create imagery. And so what happens is you actually get a very detailed image of the brain. So more detailed than the CAT scan. It doesn't require X irradiation, and you can see brain slice images in any different angle. So this can be used to generate a detailed image of the whole brain. So you can go from the anterior to the posterior, and you can see different angles and different views of all the slices. Diffusion tensor imaging, or DTI, enables visualization of large bundle of axons in the brain through the fact of measuring diffusion of water. So water diffuses much more readily alongside axon membranes than across them. And so looking at this difference, it can be used to detect axon bundles. And then you can actually trace these axon bundles and look at the different levels of connectivity between regions of the brain. So myelin sheath, which we talked about already, oligodendrocytes and Schwann cells. Hopefully you haven't blocked that out yet. But axonal membranes are encased in myelin, and myelin is white matter. And so this diffusion tensor imaging is really a great tool to be able to look at the connectivity between regions of the brain, because those are axons, axonal fibers that are connecting different regions of the brain, where you have connections from one area, soma, making a synapse onto another cell in a different region of the brain perhaps. fMRI, along with positron emission tomography or PET scans, are functional. So these are functional. Meaning, I can put you in an fMRI, or I can give you a PET scan, and I can ask you to do a task, and I can look at what area of the brain is lit up and what area of the brain is functional during that given task. So the basic principles of fMRI are we're detecting changes in blood flow and metabolism. So if you think about what your neurons and what your brain cells need in order to survive, they need glucose and oxygen, which is going to be found in the blood. So more active neurons demand more and more glucose and oxygen. Thus, more blood needs to flow to active regions of the brain when doing a particular task. So what happens with fMRI, as well as positron emission tomography, is it lets us look at changes in blood flow that will reveal brain regions that are more active under different circumstances. So if I were to ask you to look at very violent images, I might see increased activity in the amygdala, which is important for emotional tagging of memories, also the emotional characteristics of images, etc. And one last thing on positron emission tomography is this has to deal with injecting of radioactive glucose. So, I mean, if you were thinking about the difference between PET scans or fMRI, fMRI might be a little bit more advantageous if you don't really like the idea of being injected with something radioactive. Now, it does decay over time. It's not just staying with you, causing you to be radioactive. But both of these tasks are allowing us to obtain functional data. So here are just some review terms for neurons. So gray matter is the matter in the brain that is composed of neuronal cell bodies and dendrites. The cortex is any collection of neurons that form a thin sheet. This is Latin for bark. The nucleus, you can think about a distinguishable mass of neurons that are found usually deep within the brain, like the lateral geniculate nucleus, which you'll come to love when we start talking about vision. It's a cell group in the brainstem that relays information from the eyes to the cerebral cortex. The substantia, this is a group of related neurons deep within the brain, but usually with less distinct borders than those of nuclei, for example, the substantia nigra. Locus, which is a small, well-defined group of cells, and ganglion, a collection of neurons in the peripheral nervous system specifically. Now, whenever I use these terms, I'll use them in context, so I'll definitely point out when we start talking about the substantia nigra and the red nucleus, regular cortex, gray matter versus white matter. We'll go through these as through time. And some review terms for axons. You have a nerve, which is a bundle of axons. So like the optic nerve is a bundle of axons, ganglion cell axons from the retina, precisely. White matter is the generic term for the collection of central nervous system axons. A tract is a collection of central nervous system axons having a common site of origin and a common destination, such as the corticospinal tract. A bundle is a collection of axons that run together but don't necessarily have the same origin or destination. A capsule is a collection of axons that connect the cerebrum with the brainstem. A commissure is a collection of axons that connect one side of the brain with the other. And a lemniscus is a tract that meanders through the brain like a ribbon. And again, as we go through each of these terms through the course, they'll become more clear. Now, I'm not going to give you a question or anything like that that is saying differentiate and tell me all the differences between all these collections of axons, and no. You'll be able to learn these terms in time as they relate to actual structures, like the medial forebrain bundle, the optic tract, white matter that is composed of myelinated axons versus gray matter. So now we're getting into the formation of the nervous system itself. Sorry, I'm grabbing. Usually whenever there's like a pause, it's because I'm drinking something like some water or some tea or something. So during the period known as gastrulation, this is an important period. Without gastrulation, you do not exist. The developing embryo will reorganize to form 3 germ layers. This forms the ectoderm, which will give rise to the skin and nervous system. This will be important to know. Mesoderm, which is the bones of the skeleton and muscle, and endoderm, our digestive tract and organs. So during the period of gastrulation, the developing embryo reorganizes into these layers. These layers are what make you, right? So in development, your ectoderm is going to give rise to skin and all of the nervous system. The notochord, which is a cylinder of mesoderm cells, actually sends inductive signals to the overlying ectoderm, and these inducer molecules are called neural inducer molecules. They essentially tell the overlying ectoderm to become neuroectoderm specifically. Now, why is this important? Well, here's the ectoderm, and here's the mesoderm. This is the notochord, which is the cylinder of mesoderm cells. This is ectoderm, and then this is mesoderm. If you didn't have something tell the ectoderm to become skin versus nervous system, what would happen? Well, there would be some incredible dysfunction, and you probably wouldn't exist. So a part of the ectoderm that will become nervous system is known as the neuroectoderm. This is due to the fact that noggin, chordin, and follistatin, which are neural inducer molecules, are actually released from the mesoderm and actually go to the ectoderm and tell the ectoderm, specifically that will receive that information, to become nervous system or to become neuroectoderm. The area of ectoderm that does not receive this signal is going to be bound to become skin. Now, during a period known as neurulation, physical changes begin to occur within that neuroectoderm portion. So the neuroectodermal precursor cells will thicken into a neural plate. The ends of the plate will fold inward to form the neural tube, and the neural tube will give rise to the central nervous system. So the entire central nervous system develops from the walls of the neural tube. It also gives rise to most of the peripheral nervous system. Now, the neural crest, which is tissue that is found at the border of the neural tube, and the non-neural ectoderm, gives rise to the production of our peripheral nervous system cells. So all neurons with cell bodies that lie in the peripheral nervous system derive from the neural crest, so our sensory neurons. The mesoderm on either side of the neural tube will form bulges called somites, and something should be clicking in your head right now that this is related to our somatic nervous system. The skeletal and striated involuntary muscle develops from these somites. And check this out, mesoderm, bones of skeleton and muscle. So here, the notochord, which is a small bit of mesoderm, sends inductive signals up to the ectoderm to tell it to become neuroectoderm. You have folding occurring, the neural plate thickening, folding occurring, causing it to create a neural tube, neural crust, and then the area that did not receive those neural inducer molecules is the epidermis, or the skin. So here's an electron micrograph of this occurring right here. So your entire central nervous system, and if you think about the central nervous system, it's essentially a tube. Now the brain gets a little bit more complicated, but if you think about it, the basic idea through the spinal cord going up into the brain is a tube. So the formation of the neural tube, again, you have the mesoderm sending inductive signals up to the ectoderm above it. That'll be neuroectoderm. Thickening of the neural plate, folding to cause formation of a neural tube. Somites giving rise to the somatic nervous system, and then the neural crust and the neural tube. Differentiation is the process by which structures will become more complex and functionally specialized during the period of development. So you didn't just stay that nice little tube. So the first step in development at the rostral end of the neural tube of the three primary vesicles. So the rostralmost vesicle is the prosencephalon, the middle vesicle is the mesencephalon, and the caudalmost vesicle is the rhombencephalon. And these are also terms that refer to just divisions of the brain. So if you're talking about the forebrain, this is also known as the prosencephalon. If you're talking about the midbrain, this is mesencephalon, and the hindbrain is known as the rhombencephalon. So these neural inducer molecules, nogancordin and follistatin, send signals to cells in the anterior neural tissue. This allows for the promotion of the development of forebrain structures. Retinoic acid, on the other hand, sends signals to cells in the posterior tissue, and this helps to promote the development of hindbrain structures. So here, along the level of the neural tube, you're seeing differentiation where some regions of the tube will become prosencephalon or forebrain, some will become mesencephalon, and some will become hindbrain or rhombencephalon. So secondary vesicles sprout off on both sides of the prosencephalon or the forebrain. These are optic vesicles and the telencephalic vesicles. So here are the telencephalic vesicles, which are giving rise to our hemispheres and our cortex. And here are our optic vesicles here. So the diencephalon is the central structure remaining after the secondary vesicles have sprouted. So the retina and the optic nerve are actually part of the brain and not the peripheral nervous system. They arise from the brain. So the prosencephalon, or the forebrain, is made up of the telencephalon and the diencephalon. The diencephalon is going to be composed of the thalamus and the hypothalamus. So the telencephalon are cerebral hemispheres, the olfactory bulbs, and the basal telencephalon. And the diencephalon is composed of the thalamus and the hypothalamus. The thalamus is a sensory relay station for all the senses except for one, that's olfaction, or your sense of smell. Now, you do have nuclei that lie within the thalamus that do convey information about smell, but it's not critical for smell information to go in through the thalamus before it goes to the primary cortices. The hypothalamus is also in the diencephalon, and this is crucial for our control of visceral or autonomic nervous system functioning, so helping to regulate body functions in response to our needs. So the hypothalamus is often thought of as the 4 F's: fighting, fleeing, feeding, and fornicating, or another F word that you want to use. Major white matter systems exist within the brain, helping to extend from developing forebrain areas to other parts of the nervous system, making connections. You have cortical white matter, which contains all the axons that run to and from neurons that lie in the cerebral cortex. The corpus callosum, which is an axonal bridge that connects the two hemispheres together, the left and the right. And the internal capsule, which helps to link the cortex with the brainstem, particularly the thalamus. So the cerebral cortex helps to analyze sensory input and command motor output. And the thalamus is a gateway to the cortex. Again, all the senses except for olfaction go to the thalamus before being sent to the cortex. So this is a general shaping of our sensory systems, and we'll talk about this more when we go into the next chapter, which is chapter 8, where we'll start talking about olfaction and gustation. But in general, this whole principle of our sensory systems exists. So you have your eye, your ear, and your skin in this example going up in their sensory neurons going up to the thalamus. Information will be received in the thalamus. From those representative nuclei, you'll have information sent to the cerebral cortex. Now, the cerebral cortex is where our primary motor or our primary cortices lie. So for the eye, this is primary visual cortex. For the ear, you have A1, which is primary auditory cortex. For skin, you'll have S1, which is primary somatosensory cortex. And this is where you're going to get the beginning of that sensory perception. So the forebrain is involved in perceptions, conscious awareness, cognition, voluntary action. This is what makes us really human. So it's dependent on its connections with the brainstem and spinal cord because, again, if you had the will to move, but you didn't have the spinal cord connections to the muscles, you physically couldn't move no matter how much you willed it. So the cortex receives sensory information, helps to perform or form perceptions of the outside world, and will command voluntary movements. So it receives sensory information from the thalamus, which must reach the cortex for conscious awareness. Now, movement is through projections to the brainstem and the spinal cord, and the basal telencephalon, which is made up of the basal ganglia circuits. And you'll learn a lot about the basal ganglia circuits in 376. Now, the cortex in the level of the neocortex also includes the hippocampus, which is involved in learning, memory, and emotion. The basal telencephalon are structures that are deep within the cerebrum, where the range of functions vary and communicate extensively with the cerebral cortex. Now, the ones that we often include in this are the basal ganglia, which are important for voluntary movement, septal nuclei, important for our reward and reinforcement of stimuli, the nucleus basalis, which is important for acetylcholinergic neurons, implicated in Alzheimer's disease, as well as the amygdala, involved extensively in emotion and fear. Now, as we move to the midbrain, we have further differentiation. So you have, in the midbrain, you have the tectum, which is the dorsal surface, and the tegmentum. So the dorsal surface, or the tectum, will lie above the tegmentum. And the cerebral spinal fluid filled space that lies in between is known as the cerebral aqueduct. And this is a good landmark for identifying where you are in the brain. If you know where the cerebral aqueduct is, then you know that you're within the midbrain. So midbrain structures contain axons descending from the cortex that will then go to the brainstem and the spinal cord, such as in the corticospinal tract. So this is an information conduit from the spinal cord to the forebrain and vice versa, as well as through sensory systems helping with control of movements. The tectum is made up of the superior and the inferior colliculi. The superior colliculus receives sensory information from the eye, and the inferior colliculus receives sensory information from the ear. So visual information is going to go through superior colliculus. Auditory information will go through the inferior colliculus. In the tegmentum, you have the substantia nigra, which is dopaminergic or dopamine cell bodies. In Parkinson's disease, you see a degeneration of those dopamine cells, as well as the red nucleus helping to control voluntary movement in the tegmentum. So other cell groups within the midbrain have axons that project widely throughout the central nervous system and help to function to regulate consciousness, mood, pleasure, and even pain. The hindbrain, or the rhombencephalon, contains the cerebellum, the pons, and the medulla. Now, the cerebellum and the pons are collectively referred to as the metencephalon, and the medulla is known as the myelencephalon. The cerebellum is important for movement control as well as aim, rhythm, and timing. So if damaged, this can lead to uncoordinated and inaccurate movements. Usually, hopefully none of you have ever or will ever be pulled over for drunk driving, but if you were, the police often use the finger to nose test, where if you extend your arm out with your finger, your second digit or your pointer finger pointed out, and you're asked to place your finger on your nose, oftentimes drunk people really can't do this because the cerebellum is one of the first areas of the brain that are affected by alcohol. And if you've ever been drunk, I'm pretty sure you can recall that you probably don't walk in a straight line all that well. The pons acts as a switchboard, helping to connect the cerebral cortex to the cerebellum. And the cerebellum and the pons are again referred to as the metencephalon. So these are the metencephalon. The medulla controls vital functions. So again, most of which would result in death if it was damaged. So the medulla composes the myelencephalon, and it also contains cochlear nuclei, which help to project axons to different structures such as the inferior colliculus, playing a role in hearing and audition as well as vestibular systems. And then decussations exist here as well, where decussations are crossing of axons from one side to the other. And this is known as the pyramidal decussation, particularly with the medullary pyramids, which is a major white matter system in the medulla. So this is the differentiation of our rostral hindbrain. The rostral hindbrain is our metencephalon, or our pons and our cerebellum, and our caudal hindbrain is our medulla and the medullary pyramids. So this is what I was referring to as a pyramidal decussation, where neurons in the motor cortex, and this is the brain of a rat. And notice again how we don't have a lot of the convolutions that exist in the human brain because a mouse and a rat are very, very simple creatures. But a neuron in the motor cortex, you'll have information that will cross over in the level of the medulla at the pyramidal decussation and go to the opposite side to control motor commands on the opposite side. So the forebrain, also known as the prosencephalon, is all of this. This is made up of the telencephalon, which is the cortex, and the diencephalon, which is our thalamus and our hypothalamus. The midbrain, which is made up of the colliculi, as well as the tegmentum. The metencephalon, which is our pons and cerebellum. And then the myelencephalon, which is the medulla. So the hindbrain is here, midbrain is here, forebrain is all of this. Now the upper part of the butterfly wing, if you were to take a cross section of the spinal cord, you see this generally nice looking feature where it looks like butterfly wings. The upper part of the butterfly wing is the dorsal horn, and the lower part is the ventral horn. Now again, if you remember the Bell-Megendi law, you're probably thinking and you probably know what things are coming in and what things are leaving each of these horns. The dorsal horn is where sensory information is coming in, ventral horn is where the motor information leaves. And the gray matter that is found between these horns is called the intermediate zone. So everything else is white matter, where there are essentially columns of axons running up and down the spinal cord, taking information from the spinal cord, taking it to the brain, and sending information from the brain down to the spinal cord out to the periphery. So you put all the pieces together. So note how there are differentiation. It's not just here's a tube and then the brain comes about. You have differentiation where each individual region within a given region will further differentiate to give you certain structures. So you have the prosencephalon, which is made up of the telencephalon and diencephalon, the mesencephalon or the midbrain, and the rhombencephalon, and then the spinal cord. When you get further differentiated, you have telencephalon, olfactory bulb, cortex. In the diencephalon, you have the thalamus and the hypothalamus, the tectum and tegmentum in the midbrain, cerebellum and pons, and the metencephalon and the myelencephalon and the medulla. And then you have your ventricular system that lies within the lateral third cerebral aqueduct flowing into the fourth, and then into the spinal canal. And here are some related brain structures when we're looking at landmarks for lateral ventricles, particularly in the cortex, third ventricle, the thalamus and the hypothalamus, the cerebral aqueduct within the midbrain, and the fourth ventricle by the pons, medulla, and the cerebellum. So special features arise in the human central nervous system, and note that there are similarities. There are a lot of similarities when it comes to the organization of the brain. This is why we use other species that are lower than us to be able to study and understand the nervous system, because there's enough similarity in the brains that we can actually make conclusions regarding what would happen in the human brain. Now, of course, that's no 100% substitute for use of humans, but we can't exactly shock or do certain manipulations to humans than we could animals. So some differences arise, particularly in the size of the olfactory bulb, which I pointed out earlier, as well as the convolutions. So these are the sulci and gyri. This helps to increase cortical surface area with the more convolutions that you have. And this is where the seat of uniquely human reasoning and cognition really seems to lie. So note that in a rat, there is no convolutions. This is the cortex. Whereas in humans, you have a lot of gyri, which is like right here, and then a lot of sulci, which is here. This allows for a lot more brain to fit inside the skull, right? You have a lot of folds and grooves, which are essentially expanding the surface area. Whereas in a rat, well, you're not that advanced, right? The rat is not that advanced that it needs all that cortical surface area. And then what also is noted is the growth of the cerebral hemisphere, so the temporal, the frontal, parietal, and occipital lobes. So these are the lobes of the human cerebrum. And I'm sure at this point in your career, you have heard of these lobes before. And we'll go through some of them now. The frontal lobe lies anterior to the parietal lobe. This lobe is important for planning, working memory, personality. The precentral gyrus is also the location of the primary motor cortex, so right here. The central sulcus divides the frontal lobe from the parietal lobe. The parietal lobe is posterior to the frontal lobe, but it's anterior to the occipital lobe. Now, the parietal lobe is important for processing touch and somatosensory information. So touch information and knowing where your body is in relation to other stimuli. It also has important roles in advanced visual processing. And the postcentral gyrus, which is this gyrus that lies posterior to the central sulcus, is our primary somatosensory cortex, or S1. This is M1. Now, the temporal lobe, which lies on the lateral side of the head, or the brain, think about where your temples are. The temporal lobe is important for learning and memory. We have our hippocampi and our amygdala particularly housed within the temporal lobes. This is important for learning, memory, emotions. A1, or your primary auditory cortex, is also housed within the temporal lobe. This is important for audition. And you also have nuclei or regions of this temporal lobe that are important for aspects of vision processing. And last but not least, the occipital lobe, which is posterior to the frontal lobe, posterior to the parietal lobe, meaning that it is behind. This is important for visual processing. So your primary visual cortex resides here in the occipital lobe. So we've mentioned the cerebral cortex a few times, but let's look a little bit deeper. So there are some common features among cerebral cortices in vertebrates. So you have cell bodies that lie in layers of sheets that are parallel to the brain surface. So layer 1, 2, 3, 4, 5, 6, all parallel. The surface layer is separated from the pia mater in layer 1, and this will actually lack neurons. Now there may be some dendritic or some processes, but you're not really going to see whole soma containing neurons. And at least one cell layer contains pyramidal cells that help to emit large dendrites. So apical dendrites can form multiple branches and even extend up into layer 1. So there are several types of cortices. You have neocortex, which is divided into 6 layers. Olfactory cortex, also known as paleocortex, which can contain 3 to 6 layers. And the hippocampus, which contains 3 layers. Now, neocortex is found only in mammals. And it is this type of cortex that has really been seeing the greatest expansion over human evolution. So this is our cytoarchitectural organization. The human cortex develops into 6 layers, layers 1 through 6, where layer 4 is our main input layer. This is the major recipient of input from the thalamus, and these granule cells are the residing residents here in layer 4. Layer 1 is the molecular layer, which contains horizontally running axons and no cell bodies. Layers 2 and 3 provide intercortical processing. Pyramidal cells reside here in 2 and 3 as well as in 5 and 6. Layer 5 and 6 is our main output layer, where 5 goes to the basal ganglia, brainstem, and spinal cord, and 6 can relay information back to the thalamus. So there are thalamocortical loops, where you have the thalamus sending information to 4, which is our main input layer, then for intercortical processing in 2 and 3, then to layer 6, then back to the thalamus. Brodmann was a pioneer, particularly in our search for relation of what's going on within the brain and what is causing a particular behavior. So his idea was that each area of the brain has a distinct cytoarchitecture, and each of those areas is going to be given a number that's going to help characterize it and differentiate it from other regions. So we now know that cortical areas that look different perform different functions. So more specifically, each area of the brain has connections from a specific nucleus of the thalamus or help to project to unique areas of the nervous system. So structure is indeed related to function. So we know that our primary visual cortex is in area 17, in Brodmann area. You are not going to be required to know these numbers. But if you were studying the human brain and you were doing something that was critical for a particular area, like studying primary visual cortex, you would be seeing area 17 a lot in literature and in research. So in regards to evolution, the cortex amount has really evolved the most, not necessarily the structure. Humans haven't gained an additional brain structure that has, allowed us to be separate from our, evolutionary ancestors. It's the amount of cortex that we're fitting inside our brain, inside our skull. So we all have primary sensory areas, secondary sensory areas, and motor areas. So here in a human, you have your motor, somatic sensory area, auditory, and visual. But in other species, like a cat, it has the same areas: sensor, motor, olfactory bulbs, auditory, visual. In a rat, visual, sensor, motor, auditory, olfactory bulbs. But again, it's the amount of cortex that has really allowed for humans to explode in regards to advancement in thought and logic. So association areas of cortex are a more recent evolution, particularly in the frontal and temporal lobes, where our mind really correlates best with the expansion of the frontal cortex. So we have a lot of association cortex that isn't primary cortex, but it's doing a lot of additional calculations and additional specializations with information that we're bringing in. So understanding neuroanatomy is really important to understanding how the brain works as a whole. So neuroanatomy is the shell or the scaffold of the nervous system. And as you can see from this lecture, it's very complex. Individuals, neurologists, neurosurgeons have to understand every nook and cranny of the brain to be able to perform their jobs well. And the advent of methods to image the living brain has given a new relevance to neuroanatomy, where we're able to see in real time relations of structures to functions. So after this chapter, we have our second test, which is covering chapters 5, 6, and 7, which is our neurotransmitter systems, synaptic transmission, as well as all of this. So you will also have a quiz on just this information. So again, please read the book as well as go through this lecture particularly again. And what we will do next time in the next lecture is go over chapter 8, which is our sense of gustation and olfaction. And then we'll go into vision, hearing, and our somatic sensory system, or our sense of touch and pain. So I will see you all in chapter 8.